Okay, so welcome to this next video in which we're discussing clathrin-mediated endocytosis, where we're specifically looking at the involvement of the proteins Dynamin 1 and Endophilin. Okay, right. So far what we've seen is that when you have a target protein in the membrane that you want to endocytose, so let's go back over to our picture. This target protein will have an endocytic motif on its cytoplasmic domain. Okay, and this endocytic motif will tell the adapter proteins within the cytoplasm of the cell that this protein needs to be endocytosed. So these adapter proteins are in complexes, and the protein complex that is specifically involved in endocytosis at the plasma membrane is the adapter protein complex 2. Okay, now the adapter protein complex 2 is recruited to the plasma membrane by binding to the C2b domain of synaptotagmin here. Okay, and that interaction between the C2b domain and the adapter protein complex 2 is enhanced by the presence of the phosphatidylinositol 4,5-bisphosphate molecule within the plasma membrane. Okay, then what happens is when the adapter protein complex 2 sees an endocytic motif, it binds to that endocytic motif, and now what it's going to recruit is a clathrin triskelion. Okay, so it's going to recruit a clathrin triskelion, and then what's going to happen is that the clathrin is going to polymerize, form one of these buckyball structures, and it's going to pull the plasma membrane in with it. Now, we've discussed that what's going to happen is the clathrin buckyball formation, this polymerization of the clathrin, cannot completely form a complete buckyball and just pinch off the membrane itself, because the energy that is needed to pinch the membrane and split it in two, the clathrin polymerization just doesn't have that energy to do that. Okay. So we're now going to discuss the protein that is responsible, well, the proteins that are responsible for that pinching off, and they are Dynamin 1, so Dynamin 1, and Endophilin. So Dynamin 1 and Endophilin are what we're going to discuss now. Endophilin. Okay, so we'll begin with Dynamin 1. Now, this harks back to what I said right at the beginning of all of these videos on endocytosis. So the first video on endocytic motifs, what I said was that endocytosis was dependent upon calcium level. Okay, so let me draw an axon terminal here. Okay, so we know that the exocytosis process is dependent on calcium. So if we've got a docked synaptic vesicle here, then we know that when an action potential propagates along the axon into the axon terminal, this causes voltage-gated calcium channels of the N and PQ type to open, and they cause calcium level to go up within the cytoplasm of the cell. This then leads to the fusion of the synaptic vesicle membrane with the presynaptic membrane, i.e. it leads to exocytosis. So increased calcium level within the cytoplasm leads to exocytosis. However, it also produces endocytosis. Okay, so when the calcium goes up, it also will trigger the beginning of endocytosis. So it will trigger vesicles to actually be pinched off, and it will trigger the endocytosis process. And the way it's going to do this is it's going to activate this Dynamin 1 protein, and we're going to see how. Okay, right, so basically all of this process we've seen so far, the budding off of this vesicle by the clathrin polymerization, this is calcium independent. This will happen without calcium. So basically, you have in your cell membrane lots of these budded vesicles which are surrounded by clathrin coats. So let me draw them surrounded by clathrin coats, but which cannot complete the endocytosis process until they get the calcium signal, because the calcium signal is necessary for the final stage in the endocytosis pathway, i.e. the stage where you pinch off this vesicle from the plasma membrane and separate it into an actual endocytic vesicle here. And that endocytic vesicle will still be coated in clathrin. So let me coat it in clathrin. But that step from here, from one that's just a bud in the plasma membrane to an actually separate clathrin-coated endocytic vesicle, that requires the calcium signal. And we now want to look at why. Okay, so when calcium goes up in the cytoplasm, it's going to bind to the protein calmodulin. Now, calmodulin is one of these proteins that everyone's heard of, but very few people actually will be able to tell you anything about it. So, I want you to be one of these people, so I'm going to tell you something about calmodulin so that you actually know what it is. Okay, so, calmodulin, then, is a protein that in cartoons is often drawn like so. It has two lobes, okay, known as the N lobe and the C lobe. So we'll call this the N lobe here, and we'll call this one here the C lobe. Now, both of these lobes have two calcium binding sites. Let me show these here. Two calcium binding sites sitting next to each other. Okay, so a calcium combined in here, a calcium combined in here, calcium combined in here, a calcium combined in here. So overall 4 calcium cations combined to calmodulin. Now these calcium binding sites are what are known as EF hand domains. Okay, and I want to explain to you, or give you at least some sort of an explanation as to what an EF hand domain is. Okay, so let's do that. So an EF hand domain then. An EF hand domain is a structure within a polypeptide that is capable of binding to calcium, and generally they look something like this. Okay, so if this is the polypeptide strand here, this line represents the polymer of amino acids. So you've got amino acid after amino acid after amino acid after amino acid. This is the polymer of amino acids joined via peptide links. So they fold down like this and form this loop, and this is an EF hand domain. Now, the amino acids that are in the polypeptide within this loop here, generally they are acidic amino acids. So they have acidic residues, they have acidic R groups, okay? Now, you might say, well, aren't all amino acids acidic? Amino acid. But remember, the carboxylic acid group of the amino acid will be involved in the peptide bonds, so it won't be still in the native acid structure. So yes, amino acids are all acids, but well, it's actually debatable as to whether they're acids, because the amino group is actually a base, so you've got an acid and a base there. But the point is, the carboxylic acid group of the amino acid group of the amino acid isn't even going to actually be in its normal structure, because it will be polymerized in the polypeptide. So, instead, what I mean by acidic is that they have an acidic R group. So, let me give you an example of an acidic R group, and let me try and help you to understand why this is going to help us bind calcium in this loop. Okay, so, let's take the example of aspartic acid, and I'll show it as though it's bound in a polypeptide. So here's the amino group bound to the carboxylic acid group of some other amino acid. Here's the alpha carbon of the amino acid, and here's the carboxylic acid group, again bound in a peptide link, so it's not still as a carboxylic acid group, instead it's as an amide. Okay, so the R group then of aspartic acid. Aspartic acid has a methylene group, and then it has a carboxylic acid group here. Right, so this is aspartic acid. Aspartic acid, the single letter amino acid code for which is D, just to add a bit of extra information. Okay, right, so this is aspartic acid. Now, why is it an acid? What's the definition of an acid? An acid is any molecule that's capable of donating a proton. Okay, now this molecule is an acid because this hydroxyl group, this alcohol group within the carboxylic acid group, is capable of breaking that proton off, basically. So, if I draw the structure out again down here, here's the carboxylic acid group here. The oxygen is bound to the hydrogen like this, and there's one electron provided by the oxygen and one electron provided by the hydrogen atom. Okay, now, what can happen is you can break this bond, and you don't break it evenly. If you were to break it evenly, what would happen is the hydrogen's electron will go back to the hydrogen, and the oxygen's electron will go back to the oxygen, and you create two free radicals. That's not what you do. Instead, you basically give both of the electrons to the oxygen. You break it like this, and none of the electrons go to the hydrogen. So hydrogen goes off. And all that's left of the hydrogen now is the hydrogen nucleus, which usually is just a proton. Occasionally it will have some neutrons in as well, but the most common form of hydrogen, the most common isotope of hydrogen, is to just have one proton. Okay, so you've now donated a proton, and therefore you are an acid. Okay, now what do you get left over? Well, you get this carboxylic acid group now with an oxygen that has a negative charge on it because it's gained this electron from the hydrogen. The hydrogen nucleus, the proton, goes off with a positive charge, and the oxygen is left with a negative charge. Now, the structure that you're left over with, the aspartic acid where you've got this carboxylic acid group where the oxygen has a negative charge and no proton, this is known as the conjugate base of aspartic acid, and it even has its own name. The conjugate base of aspartic acid is called aspartate. Okay, so that's the strict difference between aspartic acid and aspartate. People use them interchangeably for good reason, but the strict difference between the conjugate base of aspartic acid, strict difference between aspartic acid and aspartate is that aspartate is the conjugate base of aspartic acid. It is the molecule when you have lost the proton and now have a negative charge. Okay, now why do people use them interchangeably? Well, basically, if you have 100 aspartic acid molecules, if you start off with 100 aspartic acid molecules, a certain fraction of them will donate their protons away. So, when you actually have aspartic acid molecules in a solution, not all of them will be as aspartic acid. Some of them will be as aspartic acid, but loads of them will be as aspartate. So you'll have a mixture of both of them. So you can really use their two names interchangeably. There's no more truth really in saying that you've got aspartic acid in a solution than you've got aspartate in a solution. Okay, so that's why they're used interchangeably, because if you have loads of these molecules, some of them will be protonated and some of them will be unprotonated. Okay, right, and the reason it's called the conjugate base of aspartic acid is because this structure is actually no longer an acid, it's actually a base. It can't donate a proton anymore. Instead, it can actually receive a proton, and the definition of a molecule, well, the definition of a base is a molecule that is capable of receiving a proton, so this actually is a base. Aspartate is a base. Okay, now, if you put loads of these acidic amino acid residues in this loop here, and there are more amino acids that are acidic, not just aspartic acid, glutamic acid, for instance, is another example. If you put lots of them in here, then some of them will still have their protons, some of them will be in the protonated form, but a bunch of them will give up their protons and will go into the conjugate base form. And in the conjugate base form, they have a negative charge. So basically, by putting a load of acidic residues here, some of them will donate their protons and gain a negative charge. So you've actually put loads of residues that have negative charges here. And that's perfect for coordinating a divalent cation like a calcium cation. Okay, so that's what an EF hand domain is. It's one of these polypeptide domains that's capable of binding a calcium ion. Okay, now, EF hand domains rarely go around on their own. Instead, usually, in a polypeptide, what you'll have is instead an EF hand domain, and then next to it, you'll have another EF hand domain here, okay? So basically you have a dimer of EF hand domains next to each other, and this is known as an EF dimer. Okay, now, each of these loops is capable of binding a calcium ion, so you can put a calcium ion in this EF hand domain, and calcium ion in this EF hand domain. So basically you've got two calcium binding sites next to each other. This sort of a structure is what you have in Calmodulin. You have an EF dimer in each of these two lobes. You have an EF dimer in the N lobe and an EF dimer in the C lobe, and that's how you get these two calcium binding sites next to each other. Right, so at the moment, this structure that I've drawn here is the Calmodulin when it's got no calcium bound to it. And there's a special name for that. It's called apocalmodulin. Okay, so apocalmodulin refers to Calmodulin when it has no calcium bind to it. And basically, you can see that I've drawn it's sort of in this hunched over shape, basically, with the N and the C lobe quite close together. In addition, this linker here between the two lobes, this is a linear polypeptide. This is going to change when calcium binds. Apocalmodulin is also referred to as ApoCAM for short. So the shorthand for Calmodulin is a capital C, a lowercase a, and then a capital M. Okay, now, when calcium binds to these calcium binding sites here, okay, what it does is it causes a change in conformation in the Calmodulin. So the Calmodulin becomes less hunched over. Okay, so the N and the C lobe move apart, so let's have them here. And this linker between the N and the C lobe goes from being a linear polypeptide to being an alpha helical structure. So we now get an alpha helical structure between the N and the C lobe, shown here by this spring. Okay, so this is an alpha helix here. Okay, right, and now to both of these calcium binding sites here on each lobe, you've got calcium bound. So you've got 4 calciums in total bound to this Calmodulin protein now. And I'll show that by colouring in these calcium binding sites orange to denote a calcium ion binding in there. Okay, now this structure here now is known as a calcium Calmodulin complex. Okay, so this is a calcium Calmodulin complex. And the shorthand for a calcium Calmodulin complex is to call it a Ca2 plus CAM. Again, the shorthand for Calmodulin, capital C, lowercase a, uppercase M. Okay, so calcium Calmodulin complexes. So, when calcium goes up in the cytoplasm, we are going to form these calcium Calmodulin complexes. Okay, and calcium Calmodulin complexes are then going to bind to our next target, which will then act on the Dynamin 1 protein. Okay, and we'll continue this discussion in the next video.